I'm Giorgio Bertorelle and I coordinate ENDEMIXIT. This is a project started this year where we plan to use whole genome polymorphisms to study and protect 5 endangered, iconic and endemic species. 4 of them are considered good species and 1 is a subspecies. So let's see. The Marsican bear first. It's a subspecies of the brown bear, present only in a very small population in central Italy. It's 50 individuals left and isolated since several thousand years, with a genome size of 2.8 gigabases. The Adriatic sturgeon, almost extinct in the wild, with a genome size of 2.0 gigabases, but this is a tetraploid species. The Ponza grayling is a butterfly with a small genome, present only in a couple of small islands in the Tyrrhenian Sea, the Ponza Islands. The Aeolian lizard, you can find this lizard only in a few volcanic rocks and islands northern of Italy, the Aeolian archipelago. The genome size is 2.2 gigabases. And finally, the Apennine yellow-bellied toad, distributed along the Apennine chain in Italy with the northern populations with very, very small sizes and quite big genome size, approximately 10 gigabases. We are a network of 4 universities. It's me in Ferrara, and then Claudio Cioffi is coordinator in Florence, Leonardo Congiu is coordinating in University of Padova, and Marco Gerbel is coordinating in the University of Trieste. Then we have several collaborators, 4 postdocs and 2 PhD students. So which are the main goals? Yeah, first of all, we would like to produce genomic resources for this species, producing de novo reference genomes with a mixed strategies and including also chromosome sequencing for the toad, where we have a big genome size. Then we will do genome resequencing in approximately 20 to 30 individuals per species, and the final total effort in terms of sequencing will be approximately 7.5 terabytes. But the core goal of the project is to use polymorphisms to understand the accumulation of deleterious mutations, the genomic load, in small populations and in control groups. And for us, control groups will be represented by either large populations of the same species or endangered, non-endangered sister species with very, very large population sizes. We will also estimate demographic histories and use genomic simulations to predict the effects in terms of load reduction and ancestry loss of genetic rescue strategies. We will also perform a couple of functional assays. In their cell cultures, we'll try to understand the real effects of some mutations that were predicted to be deleterious and are fixed in this group. And in sturgeon crosses, we will study again the effects directly on the fitness of the offspring of some specific mutations we will predict to be deleterious. Finally, we will also disseminate our work step by step, the methods and also the results we obtain to promote the use of genomics in conservation biology, especially in Italy. Most of the samples are already available from previous studies, but we performed also some sampling last year and this year. For example, here in the first row, you can see the Aeolian lizard sampling in these volcanic rocks. And in the second row, there are some examples of the samples of the toad, the bear, and the butterfly. As I told you, we started very recently, but we have already some results. For example, the assembly of the Marsican bear genome is quite good. We have good statistics for the back biosequencing of the in the Ponza butterfly. We succeeded in the first sturgeon reproductions with the hatchling of the eggs. And in the panel, in the right bottom side, you can see some preliminary result of the functional assay in the bear cell cultures. Essentially, cells were transfected with ND5 gene with mutation typical of the Marsican bear, predicted to be deleterious. And this is a gene very important for the energy production in the mitochondria. And as you can see, the two curves correspond to Marsican bear and non-Marsican bears. So the performances here in terms of calcium uptake are different. And so probably these mutations are really affecting performances in terms of energy production. Finally, for our dissemination task, we have now a website. We have a blog in the website where we try to update as frequently as we can about our work, and we have also a Twitter account. So I invite you to have a look. Thank you very much for your attention.